Thank you, Monia. So the next speaker is Ferdinando Squitieri. He's the head of neurology at CSS Mendel Institute of Human Genetics in Rome and the head of Huntington and Rare Disease Unit in San Giovanni Rotondo. And he has had a long-standing interest in pediatric Huntington disease. And today he will address this form of HD and to address whether it corresponds to a neurodevelopmental delay or early neurodegeneration. Thank you, Ferdinando.
Thank you, Sandrine. So much grateful you invited me. Just trying to launch my presentation, just a moment. Okay, so thanks for giving me the opportunity to share our work on pediatric HD. It's a topic that is very much linked to clinics, in my opinion, and we need to understand what is the relationship between the progression of biology with the clinical developmental symptoms, and finally with the staging of the disease. I want just to remind all of you that we have so many examples concerning the relationship between neurodegeneration and neurodevelopment. There are many syndromes that are related to this phenomena. For example, think about Down syndrome. Down syndrome in people who develop Alzheimer's disease late in life, and beta amyloid with an important implication on it. So an issue related to mental retardation and developmental issues at the beginning of life become then in a neurodegenerative process. Other examples are, for example, related to Fragile X syndrome, where a dysregulation of a messenger RNA may impact with the development of the brain. And we have many other examples. One particularly important, in my opinion, is with some diseases that are similar to Huntington disease, like, for example, spinocerebellar ataxia. And one example is ataxia type 1, where a mutant ataxin alters the neural circuitry of the developing cerebellum, setting the stage for the later vulnerability of Purkinje cells in SCA1, and so many other examples. So the abnormal brain development represents an important basis for a neurodegenerative process. It's a nice paper in Science published on it. So what's happened? What is happening? What about HD? How and when the brain development may affect neurodegeneration, and then finally the clinics. What we learned so far. And we know possible anthropometric abnormalities in prodromic adult HD subjects, for example. This is the work from Peggy Nobulo's team in Iowa. These are those subjects who usually carry the most frequent mutational length of 40-50 CG repeats. However, there is a quite slight morphometric alteration, mainly in males. Such an evidence was also reported on pre-manifest gene-expanded children, those who become symptomatic later after many years, who had lower measures of head circumference, weight, and the BMI with the brain striatum of larger size than age-matched controls, thus allowing the speculation on a faster neurodegeneration process at a later stage. All these examples concern subjects with mutations in a relatively mild range of CAG expansions. As already mentioned, and has been nicely described in the article in Science by Sandrine group, an even relatively short CAG expansion repeat may affect the biochemistry of a mutation carrier fetuses with the 3940 repeats. They show the abnormal localization of Huntington and the junctional complexes that may lead to alteration of cell cohesion and neuron migration, possibly leading to heterotopia micro malformations. And this was nicely described also by Richard Hickman paper in Acta Neuropathologica, where they found heterotopia micromalformations, indeed, mainly lateralized in right brain, and whether such abnormalities are translatable into clinics is still far from being explained. And I thank very much Richard Hickman for providing this slide and for a very fruitful conversation we had some days ago. So once again, these are the first observations on the neurodevelopmental abnormalities in Huntington disease brains with the mildly expanded CAG repeats in the Huntington gene with no relationship with the clinics. So the main question remains, what is the contribution of brain development abnormalities to clinical change in the real patient's life? What best opportunity to study such a phenomenon in the most extreme pathological condition, like when HD affects kids with very toxic long mutations. This is the so-called juvenile onset Huntington disease or pediatric HD variant in particular that affects children with the pediatric age of onset and people carrying very long repeats beyond 80 CAG, like it happens, for example, in several animal models. This is a particular rare population with a severe and painful condition causing a lot of social stigma within families. Just to remind you, this is the most severely affected population with atypical presentation, no chorea, faster disease progression, shorter lifespan, most increased CAG mosaicism in blood cells, early age at onset, which may rarely occur in very young children, and sometimes before the age of 2 years. And here I would like to show you a very short video of how these very young children progress in a very short time. This is just an example of how they progress with some physical symptoms. So, what to expect in kids' brain then? A brain pattern similar to adults with just increased severity. In other terms, a smaller, reduced size of the whole brain. If you ask, and I tried, to a neuropathologist, what to expect when you handle a brain from Huntington disease, Richard Hickman told me when I handled a brain from a juvenile onset patient with a 61 repeat, my first impression was that it was so small and diffusely small. This is very different for what neuropathologists experience with other neurodegenerative diseases like Alzheimer's disease, for example, where or PIC disease, that is a typical neurodegenerative disease in Alzheimer's context that affects brain areas with some atrophic regions. So you would expect a quite diffuse atrophy in a brain from a child with a very toxic mutation. Instead, as I usually think, looks quite complex and unexpected. So what happens in a kid's brain? Kids with very large expansion beyond 80 repeats show a MRI brain with reduced striatum since the first years of life, together with the very early onset, some before 2 years of age, early gait impairment, psychomotor developmental delay, dystonic postures, and seizures. In other terms, a clinical picture that is completely different from adults with mild expansions. So these affect children of which we select the 36 highly expanded kids with mutations of over 80 repeats, confirm the data that is completely different from adults and were also confirmed by MRS data that showed reduced CAG length dependent neuron density in striatal areas in 2 kids with 84 and 200 repeats. And such abnormality was repeat dependent. Finally, even more peculiar, interesting, absolutely unique was the unexpected evidence of a preserved brain cortex and white matter in these children in agreement with a neuropathological finding in 184 CAG repeat brain. So many questions may arise from such observations. Is this a visible clinical consequence of a biological prenatal effect, like postulated in the science paper? Is this effect caused by gain of toxic function of mutated Huntington, or also contributed by loss of function of other Huntington partners? Is the striatum, the low striatum volume, due to abnormal brain development or to early neurodegeneration? And finally, what can we learn from previous studies? Here are some studies performed before the HD gene discovery, where some peculiar observations on a typical occurrence in striatum occur in islets and increased oligodendrocytes, and seem to be in line with abnormal brain development, together with the other evidence of increased oligodendrocyte population. So, and this look like an abnormality that is perfectly fit with the juvenile onset and worsened progression of the disease, for what the others stated in their papers. So, to try to answer to these questions, we launched specific programs, including several project lines, focused on clinical, retrospective, and prospective observational studies, and on biology of pediatric HD, in the attempt to detect clinical, imaging, and genetic peculiarities, which may allow the interpretation of the contribution of brain dysfunction, degeneration, and development to clinics. Pediatric HD may itself represent a human study model, in my opinion, with the potentiality to highlight clinical biological changes that we may possibly translate to the adulthood disease. This is also crucial for it to concern future therapies and the clinical trial strategies to include the youngest in therapeutic initiatives. We know this is also an EMA requirement. A rarest CHJHD is a multi-center research project funded by the Italian Ministry of Health and strongly supported by the Foundation, an enrolled HD site, to relate brain connectivity to clinics and biology in pediatric HD patients. Imaging is performed in 2 sites, Bambino Gesù Pediatric Hospital for MRI in young children, and the Catanzaro University Hospital for MRI PET scan by a new technology combining both instruments in one, only for juvenile patients with pediatric onset. These are, this is a hybrid technology combining MRI and PET scan. And let me take a moment to present our network in detail. Skin and blood tissues are obtained by the Huntington unit of my hospital in Rome. Patients are recruited into enrolled HD by the Foundation, where they are visited by a multidisciplinary team. Hannah Bakers, Willeke Van Romom, and Susanna Debot are very much acknowledged for providing neuropathological samples from brains from children, and biological samples are then processed also in collaboration with University Hospital in Rome and National Research Council. And again, finally, Sequentia Biotech is in charge of DNA sequencing. So while keep collecting and analyzing retrospectively the larger Lear Foundation amount of data, which started many years ago, we want to study pediatric HD prospectively in the attempt to track their clinical manifestations since their first years of life. What is the cognitive profile in children with very long mutations? Only general and unfocused cognitive evaluations have been performed and are available so far. Should we expect to always observe clinical features of neurodevelopmental delay in highly expanded kids? What about additional clinical features in other body districts, for example? No data available on this matter to my knowledge. What about biological mechanisms and biomarkers? They still need to be fully investigated in this population. To get answer, we need a strong motivation and a full collaboration from families. Spazio Huntington is a program from the Foundation that is named a Space for Children, where we meet families with kids at risk for Huntington, carrying within a non-medical environment and carrying a risk of HD. Special thanks also to Noi Huntington Friends, the Italian HD Youth Organization, for their constant support on communicating HD to young people. So, this first step allowed us to start a clinical prospective approach to pediatric and to juvenile onset HD. So far, we met about 70 at-risk children, despite the pandemic that is currently still limiting our work. 4 children between 4 and 12 years of age had manifest HD with genetic confirmation and the expected imaging pattern. The neuropathology from a separate cohort that was kindly provided from Lund colleagues confirms the findings that we are observing in the imaging, and shows that kids with large size mutations above 80 repeats have a preserved brain cortex or relatively quite preserved white matter, while they have a profound loss of neurons in the striatum. So this is an atypical pattern that is also confirmed by neuropathology. And a preliminary imaging performed in 2 juvenile patients with adolescent onset and relatively shorter expanded mutations of about 60 repeats by using a hybrid MRI PET scan technology showed an accelerated volume loss in gray matter with dysfunctional glucose metabolism. It is interesting to notice that these patients showed 10 times faster degenerative process than adult, that what has been so far described in adult patients. So, and you may look at the poster from Sabatini's group. It is here in the section. So, assuming an abnormal prenatal brain development in HD, these seem to be the possible scenarios we face, and this might be object of discussion. We may have a very large expansions with the loss of the neurons and striatum volume very early in life, but the brain cortex and white matter relatively preserved, at least for it to concern the structure. We may then observe large expansions with the juvenile onset, but relatively shorter than pediatric HD population, that show accelerated brain atrophy and extended loss of gray matter. And then we may have low expansions expected to start with HD sometimes in the life, with increased striatal volume many years before onset, relatively mild and later neurodegeneration. Compensatory mechanisms and biomarkers to be still explored in each of such conditions. And of course, a link between abnormal prenatal development of the brain and faster neurodegenerative process during the life to be explored. And finally, I wish to leave all of you with one more, I'm afraid, unanswered question. When are we ideally supposed to start with a treatment? And then I thank you, all of you, for the attention, and hope we may have a discussion on it.
Yes, thank you very much. So we'll move very fast to the next speaker because we are late, but I still want to ask one question because it would be bad not to ask at least one question. So what is the proportion in terms of the CAG length? Is there a strict relationship between the very long CAG length to the pediatric form, or do you find also in pediatric form a mild CAG length?
It depends on what you intend for pediatric form. If for pediatric form you intend a clinical picture that is starting with a motor and a motor abnormality, a motor manifestation, and a developmental delay, all the time these young kids show a pediatric onset. Okay. And all the time they have a very, very expanded repeat in our experience.
Okay. Okay. Thank you very much. I hope one day we can have a longer discussion in live. Sylvia.
Okay.